The next topic we'll be starting is the recovery of fermentation products, which is entitled as Principles and Operations of Downstream Processing. Downstream processing means once the fermentation is over after the desirable incubation period, then whatever the operations, whatever the processing of extraction and purification for the fermentation products is done, it is called as downstream processing, which is nothing but the recovery of fermentation products. So downstream processing is extraction and purification of fermentation products from fermentation broth. Fermentation broth will be a mixture of several things. So from this mixture of uh, solids, means insoluble substances and soluble substances, from this mixture the target is to separate our desirable fermentation product. So this process, it may be difficult as well as costly, and it is dependent on the initial composition of fermentation broth. So if it contains too many insoluble and soluble impurities, then the process becomes difficult and costly. So the aim is to obtain a higher quality product using minimum cost efficient techniques. So this is related to the fermentation economics. So in order to make the process economical, the cost of the recovery process has to be decreased, or it has to be cost efficient. What is the composition of fermentation broth which is ready for downstream processing? It contains insoluble impurities as well as soluble impurities. So intact microorganisms, cell fragments are the major insoluble components present in fermentation broth. Insoluble medium components, which are precipitated or which are the ingredients of medium which may not be utilized, as well as the metabolic products produced by the desirable organism. Now, the another point of consideration while recovering the desirable fermentation product from fermentation broth is the property of the product. So depending on the property or the characteristic of the product, we can choose a particular method of recovery or downstream processing. So these properties of the product are: the product may be intracellular, it may be extracellular, heat labile, means sensitive to heat, heat stable, it can be easily broken down by contaminating microorganisms, and its chemical nature. All these properties are very important in order to decide the methods of DSP, that is downstream processing. Say for example, if the product is highly sensitive to heat, then the care has to be taken that the fermentation broth should not be exposed to high temperature in order to avoid its degradation. For example, if the fermentation product is sensitive to contaminating microorganisms, then aseptic conditions has to be maintained in order to prevent the contamination and degradation of the product. So this is how the properties of the product are very important, and which forms the basis to decide the methods of downstream processing. So in case of the products which are intracellular, heat labile, and which are easily broken down by microorganisms, these characteristics needs to take a special care, and special equipmental assistance is required. So these factors tend to increase the difficulties of product recovery, because if it is intracellular, then the additional method of cell lysis has to be employed, so that increases the cost. So if the cells are lysed, then all the intracellular contents of that cell will be will are going to be leaked out of the cell, and that will increase the complexity of the broth. So many metabolites will be present in that fermentation broth, and then the recovery process may become complex and costly. So it increases the difficulties. Now, the choice of recovery process, it is based on the following criteria. First one is the location of the product, whether it is present inside the cell or whether it is secreted out. So whether it is intracellular or extracellular. Accordingly, we can uh, choose a particular method for purification. Second one is the concentration of the product in fermentation broth. So this is related to the sensitivity of the recovery methods. If the concentration is very less, then very sophisticated and sensitive techniques has to be used for the recovery. Third one is the physical and chemical properties of the desirable product. So these things are also important. So this will help to decide a separation procedure. Then the intended use of the product. So product can be used for generally for two reasons. One is for human consumption, second one is for animal consumption. Say for example, if it is used for animal consumption, then it has to be purified to a very high level. In case of animal consumables or applications, in case of the animals, the product may be prepared in crude form. Then the minimal acceptable standard of purity, the magnitude of biohazard of the product or broth, the impurities in the fermenter broth, and the marketable price of the product. So all these are the criteria on the basis of which we can choose a method of recovery. So in order to make this process cost efficient, the minimum, the all these properties are very important. This is in general the protocol for the harvesting of fermentation broth. Recovery of the product from the harvested fermentation broth. So it starts from uh, fermentation broth, so removal of solids. There may be or may not be cell disruption methods. In case of the product or product is intracellular, then cell disruption is required. Else the solids can be removed by filtration or centrifugation. So that will be the primary isolation of the product. Then there can be further methods of purification and concentration and the final product isolation. This is the general protocol. Now we'll try to understand principles and operations of different methods which are used in the recovery of fermentation products. First method is called as filtration. So filtration is a technique which is useful for the separation of solids from liquids. That means separation of insoluble material from soluble materials. So there is a simple filtration device that we can see, a simple filtration device which is having an assembly like a funnel shape, where there is present at the bottom there is presence of a filter cloth which is having a particular size of the pores through which the fermentation broth is allowed to pass through, and that will be the pore size will be sufficient enough to trap the solid particles. From this end, the fermentation broth is poured in, and from the bottom, so it will be allowed to pass through this filter cloth or filter paper, and then from the bottom the filtrate is collected. So as the filtration progresses, the solids will get deposited onto the filter cloth, which leads to the formation of a filter cake. That means the solid impurities. So as the diameter of this filter cake increases, the resistance to flow will increase. That means the uh, fermentation broth, the flow of fermentation broth will decrease. So in order to keep the same flow rate, there may be use of positive pressure or there may be use of negative pressure from the bottom, that is suction, or from positive pressure from the top. So the resistance to flow, the problem it can be solved by application of positive or negative pressure. So this is the principle of the process of simple filtration process. Now the major problem in this type of filtration is the resistance to flow. Means immediately if the slurry is having, fermentation broth is having high content of solids, then the diameter of the filter cake will increase and it will block the pores which are present in the filter cloth. So in order to prevent that, there may be addition of certain assisting methods. So these assisting methods are called as the filter aids that improves the rate of filtration. So these filter aids are, for example, diatomaceous earth. So this diatomaceous earth, which is a filter aid, which it can be placed on the filter cloth so that whatever the solid flux or the solid particles having greater diameter, so they can be blocked just above the filter cloth and they are not allowed to pass through the filter cloth, so thereby avoiding the blockage of the pores of filter cloth. So it can be done like fermentation broth, it can be mixed with this diatomaceous earth so that whatever the solids like cells will be trapped in this diatomaceous earth, which can be further filtered to form a moderately clear filtrate, and then that filtrate it can be passed through this filter cloth. So this is how the rate of filtration it can be improved with the use of this filter aid, that is called as Kisselger, which is a preparation containing diatomaceous earth. I hope you remember what is diatomaceous earth. Now, let us understand the principle of filtration. So, this filter aid, that is diatomaceous earth, it has a high voidage. Now, the term is very important. Voidage is the amount of filter bed area which is free for the filtrate to pass through. That means how exactly this diatomaceous earth it improves the rate of filtration, that is by increasing the voidage. Voidage means it allows the passage of filtration, passage of fermentation broth, blocking the solid particles and allowing the filtrate to pass through. And when it is mixed with the initial cell suspension, as I mentioned earlier, it improves the porosity of the resulting filter cake, leading to increased flow. There are different types of filters. Uh, Majority of them are pressure filters because the uh, major advantage of using pressure filters or application of pressures is filtration within a short period of time. So in relation to saving of time, pressure has to be applied. Now in general there are two types of filters: batch filters and continuous filters. Let us understand what are batch filters. First category of batch filters is plate and frame filters. So this is a type of pressure filter. It consists of plates and frames. So these are the two things which makes this plate and frame filter. As the name indicates, it consists of multiple plates and multiple frames within which there is presence of a filter cloth. So these plates and frames are arranged alternatively. The plates are covered with filter cloth or filter pads. So the network is like this: plate, frame. So these plates are covered with cloth, as you can see in this diagram: plate, frame, and in between there are channels that allows the passage of liquid. You can see this is the frame, and this alternative next to that frame is a plate, and that plate, it is covered with filter cloth. So a series of plate and frames are there. The flow starts like this. On industrial scale, the plate and frame filter is one of the cheapest filters, which is most frequently used in several industries. This type of filter is most suitable for fermentation broth with a low solid content. Otherwise, what will happen if it contains high solid content, then the filters may get blocked and the filtration will stop. Content and low resistance to filtration, but it is highly laborious because of high labor cost. Means the cleaning is difficult, and it is time consuming in it is time consuming process in dismantling and cleaning of the plate filters and assembling. So but nowadays we have advanced technology in which continuously the plate frame filters can work, that we'll see in the next slide. Let us see how it works exactly. You can see the red arrows here. So that shows the flow of the fermentation broth having low solid content. So it is entering into the frame. It is entering into the frame, and uh, frame is porous, and from frame it will enter into plate. While entering into plate, it has to cross through the cloth which is present around this plate. So the filter cake will develop between the two plates. That means the filter cake will develop within the frame, and the filtrate will leave this frame. It will pass through this cloth and it will enter into plate, where from there is a channel. From this channel the filtrate is collected from the filtrate outlets. This is how this plate frame filter it works. Now this is the more sophisticated diagram that you can see here, and in examination you can draw this diagram as well. You can see there are plate and frames, and plates are covered with filter cloth. The fermentation broth or slurry is entering inside the frame, and it is allowed to pass through the filter cloth, which will further enter into the filtrate will enter into plate, and the filter cake will gather inside the frame. And once the frames are filled with the filter cake, the dismantling is done, and the plate frame network is dismantled, cleaned, and then reassembled to form, which will be then ready for the next batch filtration. That is why it is called as a batch filter. This is the diagram. You can see more sophisticated diagram. The plates and frames, you can see frames, then the in between first is frame, the second one is plate. The plate is covered with filter cloth, then again there is a frame, and again there is a plate. So this plate frame will take, and this is the actual device. This is how it actually looks like. These are the plates and frames. You can see this white part, that is the filter cloth. So it is made up with stainless steel. This is the more uh, a complete diagram of this plate frame filter. So this is, you will see when you work in the industries, you will see that this is the most frequently used plate frame filter. So you need to, you will be asked during the interviews in industries, you will be asked on this plate frame filter principle and its working, because this is the most frequently used filter. This is the actual plate frame filter. You can see this device, a complete diagram. So it is a network of plate and frames, you can see here. So these plates are closed and held tightly with large hydraulic piston like this. This is how the liquid is allowed to flow through this pipe work. The slurry is allowed to flow, and this is how the slurry enters in the chamber cavity. The liquid flow, this is the filtrate which is being collected. So this is a single plate frame, we can see here. You can see alternative plate frame, and between plates have a hollow core that inflates, and that is the collection system. So this is how the uh, liquid flows. So air is allowed to pass through in order to loosen the key. So this is how the liquid is allowed to flow inside. This compressed air is responsible for the removal of the leftover slurry. Then the plates open and dry solids are discharged, like you can see at the bottom the solids are getting discharged after the loosening. So it is a complete animation that shows as the actual working of plate frame filter in the industries. Then the next one is pressure leaf filter. These filters incorporate a number of leaves, each consisting of a metal framework of grooved plates, which is covered with a fine wire mesh, zari, or occasionally a filter cloth, and often it is pre-coated with a layer of cellulose fibers, that is to avoid the blockage of the pores. The process slurry, it is fed into the filter, which is operated under pressure by suction with vacuum pump. This is in general, I have prepared a diagram. You can see the leaves, the circular disc or leaves, which are covered with pre-coat of cellulose, and at the bottom there is filter cloth. So this first arrow downwards, it shows the flow of feed in of slurry, and then it is allowed to pass through a pipe into through this leaf, and at the side there is an outlet. So likewise all these outlets of all these leaves are combined together to get a filtrate at the bottom. So these filters are totally enclosed, so it is possible to sterilize them with steam. That is the advantage. This type of filter is particularly suitable for polishing of large volume of liquids with very low solid content, or solid or small batch filtrations of valuable solids. So for the refining or the polishing process, once the fermentation broth is processed earlier in order to decrease the content of solids, then further polishing refining process it can be done by using this pressure leaf filter. So there are three types of such filters: vertical metal leaf filter, horizontal metal leaf filter, and stacked disk filter. Then the another category of filters is continuous filters. A most more popular filter, continuous filter, which is frequently used in the industries, is rotary vacuum filter. As the name indicates, it is also a pressure filter. Here there is use of negative pressure, that is vacuum, and as the name indicates, it is rotary. That means this filter is continuously operated in a rotating condition. So these are the large rotatory vacuum filters which are commonly used by industries which produce large volumes of liquid. So this is very important for the processing of very large volume of liquids. These types of filters are very useful in the industries. So uh, whenever there is a need of processing of large volume, then continuous processing is necessary, where these drum filters are highly useful. The filter consists of rotating hollow segmented drum covered with a fabric or metal filter. So it is a rotating drum inside which you can see it is hollow and it has compartments. That means it has septation, or it is a segmented drum. So there are chambers which are numbered from 1 to 16. You can see in this diagram. These are the chambers, and at the center there is application of vacuum. So the liquid is sucked inside, and at the bottom you can see uh, the horizontal line. This horizontal line is the fermentation broth. So this drum is immersed inside the fermentation broth, and it is continuously rotating in clockwise direction, that we'll see. So this drum is partially immersed in a trough containing the broth to be filtered. So it is dipped in that broth. This is the detailed diagram. You can see more improved diagram. We'll see in the next slide, of course. So you can see that this drum is assisted with strings. You can see the strings, rollers, and there is a string on these rollers, so which is responsible for the discharge of filter cake. So this slurry is fed on the outside of the revolving drum. So it is fed from the outside, as well as this drum is dipped in the fermentation broth outside of the revolving drum, and vacuum pressure is applied internally, that is at the center, so that the filtrate is drawn through the filter into the drum. So you can see that this drum is covered with a filter aid as well as filter cloth. So whenever it is sucked in, the broth will pass through this filter cloth. So solids will remain outside, and the whatever the filtrate is there, it will be sucked inside into all these chambers, and from these chambers the liquid is filtrate is collected through a collecting vessel. This is how the operation is done. The drum revolves slowly. It revolves at a speed of approximately 1 rotation per minute. The speed is very slow. So it is like this. You can see it is rotating, and you can see a discharge or the filter cake is developed on the surface. You can see here, and through this string and rolls, it will be discharged like this, like this. So what is shown in this diagram is the insoluble impurities which remains outside this drum are continuously getting discharged through this string. It is called a string discharge. So the rolls are continuously rotating, and filter cake or the impurities are getting discharged. This is the more improved diagram we can see here. The Yeah, it's very nicely shown in this animation. The rotating drum at the bottom, there is a broth to be filtered, and this is how the insoluble material remain outside and it forms the filter cake, and this filter cake is being discharged using this string like this. This is how the filter rotary vacuum drum filter is operated. So this discharge of cake, so whenever the discharge of cake is required, the internal vacuum is released, so that will cause the loosening of the cake on the filter cloth, and then it is discharged. There are three types of discharges: string discharge, as we have seen in the earlier diagram, then scraper discharge, and scraper discharge with pre-coating of drum. This is the scraper discharge. You can see this internal part towards left is the drum face, and the cake is being built up outside, and you can see there is a knife blade which is positioned, precisely positioned, so that as the drum rotates, this knife blade will cut the cake and it will not allow the build-up of cake, or the diameter, it will not allow the diameter of the cake to be increased. So accordingly this knife blade is positioned. That is called as scraper. And the third one is scraper discharge with pre-coating of drum. So only the difference is that so earlier one and this one is same. Here also there is use of a knife blade. Only the difference is that this filter cloth, it is covered with a filter aid which forms a coat. That is, for example, the diatomaceous earth. So whatever the impurities, solids which are trapped in diatomaceous earth, they can be scrapped from this knife blade while the drum is continuously rotating. So this is how the resistance to flow, it can be avoided, and the rate of filtration, it can be improved by using this type of technique. Then another filtration method is called as cross flow filtration or tangential filtration, which is responsible for improving the rate of filtration. Let us see how. So in this filtration process, in the filtration processes, as we have seen earlier, the flow of the broth was perpendicular to the filtration membrane. In contrast to that, an alternative which is rapidly gaining importance in several industries is cross flow filtration. So the advantage of cross flow filtration is that it prevents the formation of filter cakes. Remember that in perpendicular filtration process, the filter cake, as it builds up, it increases the resistance to flow. That means the broth is the rate of filtration, it decreases. So as the filter cake diameter increases. So in this cross flow filtration, the advantage is that the flow of the medium is to be filtered is tangential to the membrane, and no filter cake builds up on the membrane. That is the advantage of cross flow filtration. This is the principle of cross flow filtration. A schematic diagram, you can see this shows the tangential. So there is a horizontal filtration membrane, and the slurry is flowing from tangential end from left to right. So this flow tangential to the membrane surface, it does not allow the filter cake to be built up. That is the that is how this cross flow filtration it works. So the benefits of cross flow filtration are efficient separations. It is a closed system and there is no addition of filter aid, so that decreases the cost of filtration. This is a diagram that shows the flow sheet for this cross flow filtration. You can see this is the chamber, then from bottom there is a pump that allows the passage of slurry from this chamber to the membrane filtration membrane pack, which is tangentially arranged, and then the retentate will be collected and it will feed back into the storage tank. So this is how the pump starts. When the pump starts, it will take the liquid from the storage tank to the membrane filtration pack, which contains the membrane filter. Then the retentate, the remaining liquid, is it will go in upward direction, that will be called as the retentate containing the solids, and it will go back to the storage tank, and the filtrate is collected from the bottom. So this is how the system works. After filtration, the second method is centrifugation.